walking into the water, a wade in and seeing whether you can see his sneakers. It's the same idea, but we're doing it in the middle of the river, in the middle of the creek. So we lower this black and white pattern. It's called a Secchi disc, and we lower it into the river, and then we pinch it off at the very top, right when the white and the black disappear. We pinch off on the string, and then we pull it up and we measure it, and that gives us our clarity reading. And obviously, the better the clarity means more sunlight's coming in, and the more sunlight that's coming into the river means there's more ability for the underwater grasses to thrive. And it's just like trying to grow tomatoes in a forest, folks. If you don't have sunlight, the grasses won't grow, right? So that we have good clarity is encouraging this rebound of underwater grasses. And this was a record, folks. Look at this. This is the clarity up at Little Round Bay. This is 2.36 meters, and that's what that looks like. I mean, that's absolutely amazing. So, you know, I'm thinking this is what we're seeing on the river as we do it, and then yet our grades come out and they're not quite as good. And depending on where you are, the grades are terrible. But anyway, this is our field investigator, Izzy Ketchum, and our crew member, Evan Rockenbach, helping us out yet again. So here's some of the grades that we came up with for water clarity. These grades were developed using the guidelines given to us by EYCs, the same group that just released their data. And you can see here that in the main stem of the river, these bolded areas, that's where we had our best reading for clarity. And then you'll see way up here at the top, Indian Landing got a D. It was horrible. And the reason for that is Indian Landing is right at the mouth of the Severn Run and all of our headwater creeks. And despite what we would like to think about our non-tidal creeks up there, they are really in terrible, terrible shape. They're just dumping mud and sand into our river constantly. So at Indian Landing, you're seeing the direct impacts of the poor state of affairs in the headwaters. It just obliterates vision up there. It's terrible. Then you can see here, most of these dark points here, most of them are in the main stem of the river. You can see the middle creeks here are starting to fall off as you get deeper into the creeks. And then the good news for our oysters, we're getting C plus where our oyster reefs are. The folks down in Annapolis Roads and Bay Ridge, unfortunately, in Lake Ogleton, just horrible clarity there. They're getting overwhelmed with stormwater runoff, and you can see it right there with those kind of grades. It does look a little better across the river over in the Whitehall Creeks. Not quite so bad. This D here for Mill Creek, oops. Now that's because this Mill Creek is right up in the headwaters, right down. You can hear and almost feel Route 50 right there. So proximity to a highway is really bad news for clarity and the health of a river. Then we go in, the other thing we track, we do a lot of tracking on dissolved oxygen, and we're able to come up with grades for dissolved oxygen as well, thanks to the EYCs formulas. Of course, oxygen is important because fish, crabs, oysters, everything, they all need to breathe something, right? So here's another one of our volunteers. Preliminary grades, some of the grades by the region, the Whitehall Creeks had pretty good oxygen. Lake Ogleton, despite the bad levels on visibility, had pretty good oxygen levels. Our oyster reefs, fortunately, scored very well in oxygen. And this is in counter to the grade we're getting from the EYCs for the bay in general. So there's some discrepancies here. Keep in mind, we are going to each oyster reef and measuring right on top of that oyster reef to get these grades. The EYCs data that you've seen about poor oxygen in the Severn River area is based on Route 50 Bridge, which is nearby, but it's an extrapolation. So that's my best explanation for the real data we're pulling up. And this is bottom level data, by the way. There's good oxygen down there for the oysters. When you get inside the creeks, you can see Brewer Creek's not so great with oxygen. And in fact, Brewer Creek was part of the dead zone. Now, almost all of our creeks had a dead zone last year. And you can see here, this is an outline of the dead zone in the Severn River. The green area is usually where we have a normal dead zone that sort of grows a little bit every year, but it's sort of persistent. 2019, for a variety of reasons that probably Tammy could explain better than I, But anyway, we had a dead zone, and we started noticing this midway into June, and we started tracking. And you can see here, it goes all the way up to the Narrows. It covers most of Round Bay, comes into all of the creeks in the middle of the river, goes all the way down into Weems Creek, right up by that first bridge. Dead zone, dead zone, dead zone. And here's an anomaly. Look at this, Weems Creek, dead zone. Right out here under the M, that's where our oysters are. Suddenly the water, the oxygen is good there. So it's a mix. We have this good and the bad all over the place in the Severn River. Just so you all know that for a dead zone, we define that as less than 2 milligrams per liter of oxygen. And we use that, it's the same one that the Chesapeake Bay program uses in the Chesapeake Bay. And we use that one in the Severn River. We do recognize that other organisms can live with less than 2 milligrams. You can see down here, worms, they do really well with 1. The spot likes 2. Crabs can get by on 3. But look at this, clams and oysters, they want 5. Our striped bass, they want 5. So these are the things that we're really focused on. We think that a healthy level is 5 for our river. This is an example of why we're picking the 2, where it underscores why we think 2 is a dead zone level. These are the crabs that were in a live pot. There were also 5 other white perch sitting in a live trap underneath Ted's boardwalk. And we happened to monitor right near his dock every week. It's by Palisades. And the day before, we were registering dead zone water. It was less than 2 milligrams per liter. Overnight, we had a storm. And it's the storm and the wind that moved the water and the waves around. And overnight, that dead zone migrated just a few hundred yards, not even 100, but a few yards over to Ted's pier. The fish and crabs couldn't swim or walk away, so they suffocated. So that is a real life example of what 2 millimeters of liters of oxygen or less can do. So that's why we feel that a dead zone is bad. And just to show you the wide variation in dead zones from year to year, these 2 charts are for the same location in Round Bay called Round Bay South. It's just offshore from Sherwood Forest. On the left is the oxygen reading through the summer of 2018. You can see that green line there. That is our 5 milliliters per milligrams per liter of oxygen. That's the good level. And you can see last year, yeah, we had good oxygen up, and this is at the bottom, folks. This is good oxygen. Down there, you can see the red. A year later, look what happens. We have the same spot. We have oxygen down there, way down there at 0.06. This is anoxic conditions, folks. This is terrible, terrible oxygen. You can see it's way under the 2 milligrams per liter mark. And it stayed that way until late summer. And then things turned and we finally got better oxygen on the bottom there. So that's just to show you the wide variation. So you got to do this year to year to be able to appreciate what's going on here. Brewer Creek, similar situation. This is just this year when we started monitoring inside Brewer Creek. It's the first time that we've been doing all the creeks like this. And you can see for most of the summer, Brewer Creek had a dead zone at the 2 stations that we go to. And you can see how long it was. It was from early July. There is where those the lines go under the red, and it wasn't. They didn't come up again until late summer near Labor Day. Oxygen levels in Lake Ogleton. I told you earlier, Lake Ogleton got pretty bad grades for visibility because of sediment coming in from the runoff. You can see that this is a little confusing, but you can see the oxygen levels by comparison weren't so bad. They're all over the place, but they're mostly all above that red line, except one station, which is on the north branch of Lake Ogleton. That's the one closest to Annapolis Roads community. That one, that station had less than desirable oxygen levels. Okay, the good news is this absolute explosion. When I first put this together, we were estimating we had 250 acres. I've since learned that it's going to be over 400 when VIMS released its data. So this is really great news. Most of what we're finding is horn pondweed in the summer. That's what you're going to see right now. It's big, it's all over the place. And then that is slowly going to be replaced by widgeon, redhead, sago pondweed. Those are the main grasses we're finding. That's like 98% of the grass. We are also finding this thing called Eurasian milfoil. It is a non-native, according to the Department of Natural Resources. They don't want to call it an invasive in our water system because we have salty water. If you go to the Great Lakes or you go to the Adirondack Lakes, where I have been and I've seen this, and also Lake Champlain, Eurasian milfoil is a vile, vile invasive species. But those are freshwater systems. We have a brackish waterway down here, so it's not the same. Anyway, the good news about underwater grasses is that they help filter the water, they oxygenate the water, and in doing so, they're also creating habitat for fish and crabs. They provide food for waterfowl, migrant birds. We even have pictures of muskrats eating the seeds. They protect the shorelines from erosion. And I forgot to put on here that grass, just like vegetation on land, they're a carbon sink, so they do help sequester some carbon. Thanks to Jack Turner, we have this cool video of a Jimmy Blue swimming in a thick patch of, that's redhead grass right there, redhead grass mixed in with some widgeon grass. So this is really great news, folks, that you can see this. That's the good news. The picture in the middle is redhead grass because when the seeds pop through the water like that, they have a red tinge. And those are the things that the geese and the muskrats seem to snack on quite a bit. We have a volunteer Navy. They're kayak based. They go out in kayaks. You can see that they're using a GPS handheld unit, and everything we go out, we use those little plastic trays you see on the right, take samples, identify the species, and then we write everything down on a data sheet. That's really critical, to write it down on a data sheet. We geolocate every sample we take, and all this information is then uploaded to the DNR. They're able to use this to expand upon the data that VIMS is collecting on underwater grasses, at least in the Severn River, we're able to do that. So here's my theory on why we have such an abundance of SAV. We had a record-breaking rainfall in 2018, which actually started in 2017. That led to a total influx of a gigantic amount of fresh water. So that causes a salinity collapse in the Severn River. As it turns out, low salinity is a time when SAV starts to seed. It's sort of like a pine tree in a forest. They drop their pine cones, but those don't seed until a forest fire comes by. So SAV needs adverse water conditions, i.e. fresher water, to come in to really seed the place up, right? So we get this influx of fresh water for almost 2 years. And then that low salinity triggers the seeds to drop. The low salinity also brought in this proliferation of the dark false mussels. Dark false mussels are like oysters. They filter and clarify the water. Dark false mussels for the last 2 years were under the water line in the Severn River everywhere. So you can see all these little blue dots, by the way, these are all our SAV dots. This is where we were collecting data on SAV. And down here, these are where our oysters are. So they're doing their filtering, cleaning thing down here. But everywhere else, there are no oysters. But in the last 2 years, everywhere else, we had the dark false mussel. So we're claiming that the dark false mussel has helped improve the clarity at a time when we had all this low salinity come in. That means more sunlight, more seeds are being dropped, more SAV. So that's very exciting news. I'd like to wrap up with an oyster update. I mentioned that we have 4 oyster rates. We're trying to repopulate oysters in the Severn River. Our former president, Bob Whipkin, started this program in the Severn River called the Marylanders Grow Oysters. We've been doing that now for 10 years. This is a sample of these 6 to 8 year old oysters that we brought up last April on an oyster dive. This means they've survived low salinity and questionable oxygen at times and all the weird conditions that come and go. These guys have survived. So this is really good news. Yeah, I said all that, right? So we have 8 to 6 year old oyster Traces Hollow. The really good news, folks, for oysters now is that last Labor Day, we finally tracked that the salinity returned to normal. Normally in the Severn River, we should be seeing salinity levels in the 8 to 15 part per thousand range. for the last 2 years, we've had a collapse in salinity. We've barely been able to see oxygen. I mean, the salinity was in a 4 to 6 range. And when I say 6, I'm being generous. So we had this very big freshwater event that lasted almost 2 years. But now that the salinity has returned to normal and we have good oxygen, our oysters should be thriving this spring. And during the spring is their big growth season. So we're looking well. We got our fingers crossed that in 2020, we're going to restart Marylanders Grow Oysters in September. And hopefully we'll restart our Operation Oyster Reef program where we buy and dump millions of oyster spat. This is the oyster dissolved oxygen readings along the bottom. You can see that the oyster level, the oxygen level for the oysters has been up and down a lot. So what can we do to help our Severn River? Because this is what we really want to do, right? We want to celebrate kids swimming, and we want to go fishing, and we want to go crabbing, and we want to go sailing on beautiful, clean water. So the little things we can do that are help everybody can do is install rain barrels, plant trees, build rain gardens where you can, pick up your pet waste, maintain your septic system if you have one, remove invasives, and by invasive I mean the vines on land that are crowding out the underbrush in forest and wooded areas. That underbrush is a far better sponge for soaking up stormwater than some of these invasive vines. And then of course we have a ton of people to thank, all of our crew who are getting out there every week to do this. This is Izzy Ketchum, our Chesapeake Conservation Corps member. She's our field investigator for this year. She's been really a huge help. Here's some more of our pals who have been helping us out there in the year. And special thanks to the Delaplane Foundation because they've helped support us with this water quality monitoring program. Also Liz, Nathan, and Carolyn at the Chesapeake Monitoring Cooperative because they helped give us the grades. And a ton of people took great pictures. Here they are. Thank you all. Is that it?
Okay,
Sorry.
great. Thank you.
That was a race.
All right. Well, we have a couple minutes till for some questions, and I'm sure there's a lot of questions. Let's actually, John, can we maybe let folks be able to join in for and ask questions?
Yes, hang on.
Okay.
One second though. I just wanted to share one thing because Tammy put it up, but it was hard to see on her screen. And after hearing all the great stuff that they said, this is not really that important. This is the UMC Lower Western Shore grade, you know, how everything is, where things come from. And I just, you know, people couldn't see that. And Tom explaining that bridge thing is so, you know, helps understand that. But I just wanted to mention, you know, the dissolved oxygen, they actually gave a good grade. But of course, that was because they're only sampling in one place. They didn't see the dead zone, and you know.
They didn't see the dead zone because I failed to point out, but the dead zone did not reach all the way down to that Route 50 bridge.
Right.
So my point in emphasizing this is that between Tammy, Spa Creek, and the Severn River, all three of us, you know, we all have a tremendous amount of data that's not being fed into the UMC system.
Right. And that was why this presentation was so great. And coming out on the day with the UMC report, now we all understand why the UMC report is interesting, but in terms of focusing on the Lower Western Shore, what SRA and Spa Creek Conservancy is doing is so much more important and more complete.
I certainly am not trying to discount UMCs. Those guys are smarter than I am by miles. So I just want you to understand that. I'm also trying to be a homer because we've got a lot more data and a lot more incidental things that they just don't have access to yet. Yeah. But maybe next year they'll have access to all this data.
So I have another question I'm going to ask, but I'm going to hold that for later. I just, I think I changed it so everybody, if you have a question, find a space to, you know, there's a lot of people participating, so try not to step on people. But instead of sending in chat, you can send a chat to me or Virginia, or you can ask the question yourself by unmuting yourself.
Hey, this is Patrick Williams. I have a question for you. Since most of the nitrogen appears to be coming from lawns and fertilizers, you know, the phosphates and the nitrogen, Long time ago, I heard that cattails were very good to suck up nitrogen. Is there a problem with planting cattails in those areas so they become like a nitrogen trap?
You want to try to answer it, Tom?
Oh, thank you, Patrick. Unfortunately, I am not an expert on nitrogen or cattails, but I would point out that most of the, a lot of the nitrogen that we're getting is not just from fertilized lawns. We're getting nitrogen from all kinds of sources in the Severn River, including air pollution itself. So a lot of the nitrogen pollution we're getting is coming from the stormwater runoff. Now, if you could set up cattails to capture stormwater runoff before it goes into the river, that might be a great idea. I don't know anything about that one.
Yeah, that's what I was thinking. You know, like in the narrow, I live here in Murray Hill, and close to the Maryland Hall, and right there, the creek gets very narrow and muddy, and probably is getting a lot of contamination from those areas, which are very shallow and narrow. And if the cattails, they're not an invasive species, then we're going to regret planting cattails. Maybe it could be an experimental thing to do.
Well, I think the answer you get from people at the Watershed Stewards Academy or people with real understanding. I'm not a landscape architect, but
Okay.
what I've been led to believe is that any kind of a buffer that slows and absorbs the water at the stream level, like a tree is a giant sponge. Trees are great. That's the best thing you can have. But if you can have all other kinds of vegetation that slow down that water, and so that's why you're seeing on the Eastern Shore and up in Pennsylvania and other places, you see a lot of effort by agriculture outreach organizations trying to get farmers to put buffers between their cattle and their streams or whatever. It's to keep the water away from the stream itself.
Mm-hmm.
I don't know anything
Okay.
about the biology.
I just wanted to say, I think we have another question here from Ann Powers first, and then
Well,
Tara.
no, I was just going to say that I have always understood cattails were very invasive and would force out all sorts of native vegetation. And so I've seen areas where they were yanking out cattails, like Phragmites. They were similar to Phragmites in that regard. But that's just what I've heard. I'm not, that's not my field of expertise.
I would agree with you on the Phragmites part. I just don't know anything about cattails.
Right. Well, and also to add to that, you know, I agree that the worry with a lot of those is that they're going to take over. And as Tom mentioned, something that goes along with that is even if you have some cattails, a big problem is, as you mentioned, if you have these very narrow areas, that one of the things that they really want to be able to do is widen out the floodplain so that you would have multiple plants that could work, and you can really have that percolation of the water down into the soil, into a wide swath of plants. But yes, cattails, I'm not a cattail expert either, but I do know that there are concerns about the invasive nature of cattails also.
Okay, we have another question here. I'm going to ask Tara Potter. Raise your hand.
Hi.
Raise your hand like this if you want to. If you have a question,
Hi,
Tara.
and thank you for the presentation. If anybody's driven over a bridge over either the South River or the Severn River, or any of our rivers lately, the water pretty much looks like chocolate pudding. I know we've had some rain, but this seems quite unusual. Can anybody explain what's going on?
Tammy, you want to do it? You want me to yak away?
Well, there have been reports from DNR that there is a mahogany tide, but I don't think all of that. I don't think all of it can be explained by the mahogany tide. This is on Spa Creek. This is going to be the first week we're going to get out there to do testing and monitoring to really look and see what the effect is. What I've heard is that's pretty widespread, and a lot of it does not look like necessarily an algal bloom, but just a lot of suspended solid. So when we get out and get to do some monitoring, we'll have a better idea of possibly a better idea of what's going on.
Yeah, Tammy, can you just explain what a mahogany tide is for folks?
Oh, so that's a specific type of algae that totally escapes my mind now. Oh, it's right there, but it's not. I'm not going to be able to get it out of my head. That happens. That is relatively common in the area. And so when they overgrow in the winter months, when I did some research on it, it's not as common in winter months, but there are known blooms, and it seems as though it's been happening more frequently. Last winter, it was either December or January, there was definitely a mahogany tide. And it's just the specific species, specific type of algae that causes it, that causes the color to be mahogany and for it to be considered a mahogany tide.
Thank you.
This is John. I've seen it in the Tecumseh Marina, and I just actually, thanks Tammy or whoever mentioned mahogany first, maybe it was Tom. I just did a search, and today's Baltimore Sun says the largest algal blooms in 20 years have invaded the lower Chesapeake Bay and its tributaries, causing at least 2 major fish kills and discoloring the water from Sandy Point to Parker's Creek in Calvert County on the western shore. So I'm sure that what we're seeing in Tecumseh Marina is not suspended solids. I can see that it just doesn't have that character to it. And I guess we're seeing the mahogany tide in Spa Creek and at Eastport Yacht Club Marina too.
I've seen it up all over and up the river as well, and I would attribute it to be, well, I call it a red tide, but it's an algae bloom to me. But I can't prove it because I don't have a microscope.
Based on what Tammy just said, that sometimes it develops over the winter months, and given that we had such a warm winter, is it possible that that's what's led to an overgrowth of this year?
Yep, that's possible. And so what we'll probably start seeing is things like really high chlorophyll. One thing that indicates algal blooms are high chlorophyll levels. So that will, and then that causes problems later on also, because that then will also contribute to things like dissolved oxygen levels going down. But yes, temperature, again, we don't know. I don't know what the water temperature is until we get out on the water on Spa Creek tomorrow, but we'll have an indication. And if last season predicts this season, then we can expect temperatures to be higher.
Yes. Next question.
Yes, I'm Pete Galata. I just share a personal experience I had, and perhaps one of the two of you can answer my question. 2018, my wife and I took our boat from the Severn down to the Rappahannock River, and unfortunately, when I was down there, I came down with a severe case of cellulitis and ended up in the hospital trying to save my leg and my life. 8 days on IV. And the physician that treated me said that it was because of the brackishness in the water and the high temperature and bacteria. And they were treating me for Vibrio, although they weren't able to identify the bacteria. Are either of you aware of the numbers of people that have come down with waterborne disease in this area?
So this is Tammy. I don't have the exact numbers. I do know, I can look that up, but it is still relatively low. The issue with Vibrio is that that's not something we directly test for. It's not associated with, fecal contamination. NOAA has a really good model that predicts what areas of the watershed are going to have higher counts through different parts of the season associated with temperature and salinity. So it's still a relatively low number. There have been some increases. The other issue is that we all hear about it more often now. Yes, the numbers have been increasing, but it's not a large enough number that we have really good data from the population. So you were you're someone that had dealt with it.
Yes, unfortunately.
So yeah, so even though it's low numbers, it's not low enough it's not low enough. So, yeah.
When I was on the Rappahannock, it was an adventure trying to find somebody that would treat me. And it was 2 hospitals and 2 emergency rooms. And I was certain I was going to die down there in a hospital that only had 25 beds. And the doctor down there assured me that because of the frequency that they see people with waterborne illness like in Kilmarnock, they know how to treat it because of the watermen getting it.
Right.
And I don't know whether I brought it down there. It was right around the time that the Conowingo was opened up in 2018. Mm-hmm. So the water, different than it's normally been.
Did you have an exposure to, did you have an open cut? you know,
Yeah.
when they think, oh, so it was from an open cut?
Yeah, and I never thought anything of it. I never went in the water, but the doctor told me that it doesn't matter that you didn't go in the water. It could have been washing your boat down, splashing on your boat, bumping up against something on your boat that had water on it. And I never really paid very much attention to it. I've been following statistics as to the water, whether it was swimmable here, but I really never paid any attention to, a sore and sticking a Band-Aid on it and peroxide. And I left myself open for that. So I'm much more cognizant of it. And I'm absolutely mortified to go in the water anywhere on the Chesapeake. And I want to thank you both, by the way, for your work and the presentation tonight. Thank you very much.
Right. And I just want to make the point from that when you talked about swimmable. So that's one point that we always have to make, is that what we test for the water when we say it's swimmable, that really is for enterococci. Right now, there's no regular program to test specifically for Vibrio species that are that cause infection. And part of it is because even at very low numbers, because it's so pathogenic, it can be very problematic.
I think what's probably important is to go out to the public and tell people that you can get a disease like Vibrio, and that there are things that particularly boaters can do to minimize their exposure to it. I certainly, have learned my lesson, but it's something you don't want to ever go through.
What time of year was this?
This was around July of 2018 on the Rappahannock when I came down with it.
Okay, thanks.
Now you guys all curious. We're gonna have to check it out. But I think Tammy and Tom would probably vouch for this, that, when there's an extensive rainfall, everybody should be wary about getting in the water right away.
Yes, yeah, and especially from the enterococcus. The county recommends 48 hours. One of the points we make a lot is that we always try to look at this 104 CFU as the cutoff, and we show kind of red, green for good water, bad water. But you always want to keep in mind what your what your health state is. Even keeping kids out of the water is hard, right? So things like having the rule, you always shower when you get out of the water. If you have an open cut, even if the water is swimmable, if you have an open cut, you want to make sure you wash it right away. If you're immunosuppressed or immunocompromised, or you're sick with something else, then you still want to be paying attention to that, even when the water is swimmable. All of these correlations are just that, the correlations and probabilities. And as far as the Vibrio that you mentioned, that July, Vibrio tends to increase again as the water temperature goes up and the salinity. So mid to late summer is when it tends to be a larger factor in the area. I always think about when jellyfish start coming. When jellyfish start making it out, so do the Vibrio counts tend to, Vibrio presence increases.
Thank you so much.
Ursula Kuhn?
Yeah, I have a question. Well, it's a question that I'm directing to Tammy. First of all, thank you for a great presentation, both of you. But I live up on Spa Creek. I live actually above the South Cherry Grove Bridge, where they did all of the step pooling and everything. And I'm noticing a lot of Phragmites all of a sudden starting to grow up here. Should I be getting a hold of someone?
That's too bad that you have. So yes, and I'll have to look and see, because DPW and I should say the office, Eric Michelson's office that's involved with some of that restoration, an email. It wouldn't even hurt for me to send an email. Sometimes one of the things that happens for a number of these restoration projects that are put in, especially they're done by, if they're done by the county, there's not always the process put in place for maintenance afterwards. And it's not because they wouldn't like to. Part of it is time and cost. So in some situations, they do sort of rely on communities and residents to keep an eye on things. And potentially in some situations, they even rely on communities to do the upkeep. I don't know what the deal is with the step pool by Cherry Grove, but Yeah, it would be, I can even make mention to, I can send an email also to put it out there.
Yeah, it's about a block up from South Cherry Grove. It's out at the end of Southwoods Avenue.
Mhm.
I live in the Southwoods community, and I'm right up against the creek. And it's, you know, it's not right behind my house, but a little bit closer to South Cherry Grove. But it's on both sides of the creek, and I'm noticing it's just getting more and more, and somebody's got to do something about it, I'm sure.
Okay, I'm gonna write a note down about that.
Okay, do we have any other questions? I know, John, you had one you wanted to.
Yeah, I'll go ahead and ask my question to get back to the nitrogen issue, which seems to be pretty important to try to address. And I'm always hopeful that a little education can work towards behavior change. And, you know, maybe sometimes that's overly optimistic, but I was at a CERT presentation on one of the Tuesday night lectures before the pandemic hit, and it was on a stream water restoration study, and they were doing automatic monitoring, and they were monitoring nitrogen coming into the system and nitrogen coming out. And, you know, we can measure nitrogen in our creeks and in the water, and we can say, oh, the nitrogen is elevated. But wouldn't it be nice to point to, you know, this residential community, or even this area of a residential community has a really excessively high nitrogen. You know, it's not not everybody is over fertilizing, and not everybody is over fertilizing to the same level. And so it's just really a question for Tammy and or Tom is can we, you know, the sampling we do isn't generally nitrogen, but if we had volunteers to do where residential stormwater goes into our creeks, you know, point sampling during rainfall, and what's important? Do you get the first hour? Do you wait? You know, and you know, how complicated is it, and how expensive is it? And is that something we could really try to, you know, develop? And I don't know whether this would really be a follow-on meeting to brainstorm about, but that's just something I've been thinking about.
So that is a really good question, and part of it is effort and cost to track things like pulses. You know, I mentioned the term pulse. Like, there's some really nice studies, especially with things that are somewhat easier to do, like tracking things like suspended solids, like even bacteria, where you can take samples a certain amount of time when there's a storm coming, before, during, and after. And there are auto samplers that some places have access to. So that can be done. Measuring nitrogen levels is expensive, is one of the things. So it often comes out to be a cost issue. As you noticed, even on Spa Creek, we do nitrogen. We only do it every other week, and we do it at some of the sites. It requires being sent to a lab, and it's costly. It would be the best way. It's the optimal way to do that if there are areas that are of concern. We've talked in some other programs about really being able to do really storm event sampling. That's a big lift in manpower, in equipment, and in cost, and even just availability of volunteers or workers to do that, to predict when a storm's going to be ready, and then go out at all hours of the night, day, morning, and collect samples. Yes, but that would be the optimum way to do it, is to do stormwater events, time course sampling.
And I'm talking about sampling on the shore before it hits the tidal water. So at least it doesn't require a boat, but it sounds like it's still very expensive.
Right, well, and that's, so yes, so, there are programs that do that. There's a couple that down at University of Maryland, but down at Solomon's, that's a big part of their research. And they do that type of sampling, and they have grant money to do it. And they do use auto samplers. So they have areas along, they do some of their work in DC, like along things like the Anacostia, where they see some very interesting things after it's rained a lot. And they use auto samplers, and that's grant based. So there are ways to do it. It just takes a lot of effort.
John, one thing you could do, if you suspect that there's a spot there, just look at the lawn.
Yeah.
If it's beautiful green, then they're over fertilizing, and they're doing it in the spring, which we've all heard is not necessary.
I understand that, Tom. I want some data that I can show people. And in fact, I could see having competition, in fact, comparing, your block over there is worse than our block over here kind of a thing, or our block's better than your block.
Depends how big your checkbook is, John.
But the cost sounds like it's prohibitive, unfortunately. So my other question is, Tom, is you mentioned the SAV Navy. Is that people kayaking locally? Do they transport their kayaks? Do you need more Navy recruits? There may be people on this call that that are kayakers.
I would love to recruit people for our SAV Navy. And we set it up as a kayak-based thing because most of the SAV is in the shallow shoreline waters. It's up and around Round Bay, mostly. That's where 90% of it is. It starts about halfway up the Severn River. If you all know where Joyce Lane is and that day mark, that's that red mark out there, there's a light. There's a big patch of grass, and it starts there on the sunny side of the river and runs all the way up into Sullivan's Cove. And we need people to get out there and do the monitoring. Now, we have a training program. It's a fun way to use your kayak and, to get out on the water, do some science work. If you write it down, you're a scientist. It's the whole idea of a data sheet is what we use. And writing it down is how you turn anecdotal observation into science. And then we have a training session. We give you some gear. So if you guys are interested, I would love to recruit you. So just send me an email at info@severnriver.org and put SAV in there, and then we could talk later. But you have basically 6 to 8 weeks to go out and do, get 25 readings if you can do that, if you can do that much. And we try to monitor from May all the way to late September. Now, our spring grasses are dying off now, and they're going to be replaced by the summer grasses in mid-June. So there's a ton of grass to track. So yeah, I'd love to recruit people.
And if folks are interested in helping out with the water quality monitoring or SAV monitoring, they can also contact John or myself, in the EYC Green Team, and we will get you in touch with the right folks.
Okay.
Just an aside, in terms of catching those moments, one of the things our environmental committee that EYC has been doing is Tom Stalder, I think, is on this call, and actually Jason Simpson and Janice Cannon. There's several of our EYC Green Team members that are actually going out monitoring during storm events. Thanks to Bob Whitcomb's encouragement, we've been really trying to get a better understanding of the potential effect of the storm events, and make sure to capture that information. And I know our interest is also because of our oyster effort there at the marina. And Tom, it was interesting to see some of the information you were showing about the clarity of the water and the issue of obviously the role of the oysters at possibly improving the clarity of the water. We seem to be seeing pretty good water at EYC water clarity, and we're trying to get a handle on the effect of our oyster reef that we're building under our docks, and of course also our oyster conservation efforts with the Marylander Grow Oyster programs. And I know Bob Whitcomb, I see you there on the call as well. Between you and Tom, do you have any thoughts you might have about better ways we could get a handle on as to whether the oysters are making a difference?
Well, yeah, I think Tom has listened to my request for years to monitor the reef where we put the MGO oysters. And I thank you, Tom, for including that as a major measuring point and now a major reporting point. The other experiment going on the river, if you're not aware, Tom, is underneath the EYC docks is an oyster reef that's been growing for 10 years. And we would sure like to know the water quality and other positive impacts of those oysters on the waters right around the marina.
I would go out and keep data records week to week to week to week to week until you have data to prove your point, but I'd claim credit. You got to go out and get the data to prove it,
These are
though.
trends. Huh.
You know, but once you got data, you can go, yeah, look, compared to our water, and you have that nice little area there, so it's a confined area. You can say, okay, just inside your breakwater there, right? You can say whether that water is clearer than 2 blocks down the street. And, you know, that could be your reference point, but you need data to do it. Well, you
Yep.
can't hear me?
Well, thank you. And I know been talking about that, and I know we're continuing that dialogue. And we've got a team that's been trained by Tammy on how to use all of our equipment that we have, our YSI and our Secchi discs and pH meters. So hopefully we can get it out there and do some more sampling and keep. We are keeping records. You're right, though. It's we need to see the trend over time and compare it to the next door neighbors.
Yeah, that is fantastic. If you guys are going out for doing storm sampling, monitoring after storms, the more data you have on that, the better, because that's the hardest time to get people to go out, right? We go on there's Tom's picture of data. You know, we think about going out on this consistent basis because you can get people, they have a schedule, but if you have people that'll volunteer to go out, like spur the moment when there's a storm and to spend some time and do in any type of time course, that data is just invaluable.
I'd like to pick up on John's idea a few moments ago of, you know, how do we nail down the source of the nitrogen? I've been a minor voice for years, convinced that the dead zone that we've seen in Round Bay, I'm talking about the upper part of the Severn now, I'm convinced the dead zone we see every year in Round Bay is a direct result of nitrogen runoff from our septics. All of us upriver are on septic systems. I don't care how well they're maintained, unless there's a nitrogen reducing additive to your septic, you are leaching nitrogen. And that's being leached out through groundwater. It leaches through the creeks. And I think if we could institute an additional nitrogen survey up the creeks, then maybe we could nail which neighborhood is got the greatest leach.
Anybody grow up in a Catholic school and all the kids go, sister, 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 sister?
All right, Tom, you got the chair.
So Bob, brilliant idea. What we need to do, and we could do this, is we need to expand our monitoring program, the one we have now, so that we're actually moving into the fecal matter DNA category so that we could actually identify human versus animal. So we know whether it's you pooping or whether it's the geese. Right now, I don't believe we have the capability of making that determination, but we don't have any data on septic leaking out. We just know that they have a lot of septic systems. They're all over the upper river, but we don't really have any data. When we could put together a consortium of partners with Tammy and you guys and Severn River, get Tammy's school to get the funding to do the more advanced testing so we could do this, then we would have the data, we could make our point, or we could clarify it better, go, oh yeah, it's all because of the dogs and nothing else. You know, silly, but then we could finally determine it. Right now, we just don't know. So if we could just go to the step where we could actually use DNA to distinguish between which bacteria it is, Tammy could do that, I'm sure, very easily, right? All we gotta do is, all we need is money.
Well, we do. Yeah, we actually just last year got the piece of equipment that can be used to do that. Before that, we were trying to use an older piece of equipment and doing some of that.
Well, so.
And we had with some research-based stuff, we had some indications between human versus dog, but not anything that I would bet my life on.
All right, so we take what you've done and we advance it more because we now have the network to do the whole river, right? So we can take those monitorings and bring them to you, and we could actually find out and answer Bob's question.
Right, and we definitely now have the technology, and the methodology is getting much more standard, but we definitely have the equipment. And back to the
clarify, Tom.
Sorry?
I was not referring to the fecal matter that could come from a failing septic. I was referring to the normal liquid.
It doesn't matter whether it's failing or not.
Travels out of the septic, it carries the nitrogen through the groundwater, eventually into the creek waters.
By their very nature, septic systems release nitrogen.
Exactly.
So if it's failing or not, doesn't matter. If a perfect one still releases nitrogen.
So on that, I know that the county put together a work group on that. I know even Sally Horner's part of that to try to figure out about some of the upgrades and things. If it's an area where you think that the nitrogen is really high, there are kits. So there's a difference between, you know, nitrogen that's measured in open water when we're really trying to think about values to go in a downward trend and really high levels of nitrogen. There are actually some, you know, chemistry-based sort of field kits for nitrogen that are not as costly as what's needed to do that precise level of, you know, when the nitrogen is what we think of as low, but high according to the TMDL. If you have high amounts that are really coming from a point source and you have some indication it might be there, then there are some kits that would have the sensitivity to be able to do some on the spot sites to look for higher levels of contamination. So I was just thinking about that now from what you and John, from what Bob and Tom said, Bob and John said, sorry, I'll get it there, that might be worth, if there are areas there where you truly think that there is a chance that you have some reason to believe that it might be high at those points from a source. If you're close to the source, some of those kits would be precise enough, and they're not, or I should say sensitive enough, and they're not overly expensive to get a general idea, a relative idea of relative quantitative idea.
The other thing to track, of course, would be, you know, boat discharges and how serious, you know, what percentage is that? There's no data, but there's a guess. So we're developing this no discharge zone, which we could fold in this nitrogen search, the septic search, and fold in the boats and holding tanks and releases as well, and pull all that together to get a better picture of what's causing the nitrogen overload or the bacteria overload.
Well, so Tammy, you mentioned that this kit would not be as expensive because you're not looking for the low levels. So I can imagine that you have, you know, over fertilized lawns running into a creek, and now all of a sudden that relatively concentrated nitrogen relative to what, you know, you're trying to test is in the creek, and now it's a much lower level, still high, but it takes more sensitivity. Do you think a army of volunteers collecting water samples, you know, by foot in where, you know, a lot of these fertilized lawns are running off down the street, you know, collecting multiple lawns, you know, a block or whatever, down the street into a creek, collecting water samples at certain point sources that we kind of, you know, volunteers determine? Is that something that would, you know, the inexpensive kit could measure?
Potentially, yes. So there are several companies that have kits. In our lab, we do some semi-sensitive tests, which are definitely more sensitive than the things that you take out in the field, and but not as sensitive as what you get from something like these auto samplers that like EYCs has. But the same company that we get some of our reagents from for mid-level testing has some like field test kits for nitrogen. The sensitivity, again, it would have to be a reasonable spike. So I'd have to look into it a little bit to see, you know, how close you have to be, what you expect to get from a septic before you just want to jump in. But those field kits are getting better. And instead of like looking at like midstream when it has been diluted and looking at the overall picture, they're getting better to find some significant contamination. So it's a possibility. The kits usually run anywhere from $50 to a couple $100 for a, you know, something like 20 tests, and they're quick, and they're relatively quick too. So yes, in that case, then if an army of volunteers who want to go look, there would be a possibility for that.
Could have official monitoring day.
Yeah.
Well, I hate to cut things off, but I think we've gone we usually try to wrap everything up for sure by 8:30, and we're there. But I do think this is a good discussion. Tom, I know you were raising your hand. You wanted to say something about this idea.
I'll spare everybody. That's okay.
I think all of us really are quite intrigued. So I think Tom and Tammy and John and you, I think we all maybe ought to have offline powwow later on about this idea and explore it a little bit further, because we do have a good group of volunteers, I think, at Eastport Yacht Club that are really interested and concerned about our communities and would like to get a handle on where the hot spots are, the problem areas are. And so I think there's a good set of volunteers that we could potentially recruit for such an effort. Before we close out, I did want to just ask our Commodore, Liz Filter, do you want to add anything, or?
I just have enjoyed this immensely, and I just can't thank Tom and Tammy enough for sharing their knowledge and also their time. But I also, you've now sent me down a new rabbit hole with the Chesapeake Data Explorer. So I'm going to be spending a little time on that, and I appreciate that. And I also appreciate just always reminding us those little things that we can be doing in our own lives. So we can volunteer at EYC, but then also those things that we can do at our property. So I thank you for that. And of course, Virginia and John, again, being able to deliver this kind of content to my house. Thank you. I really appreciate that. And the entire environmental committee and all the work that they do year round. I have to tell you though, I'm wondering, Virginia, where's your Secchi disc? You always show it up and it's always within arm's reach. I know that every annual meeting, you always bring that with you. So I'm wondering, it must be hidden in your pocket right now.
No, it's actually right now down at the shed. You know, they're on CDOC, the yellow shed.
I appreciate everybody's commitment to this, and I look forward to more programming like this. So thank you all so much.
Well, thank you, Liz, and for joining us. And also, I did want to just recognize David Barker, who's the current president of Severn River Association. David, thanks for joining us. Do you have any parting thoughts about the discussion tonight? I'm putting him on the spot here, and he's probably muted.
Unmuted, and I want to thank Tammy. She's a wonderful colleague, and Tom, and especially you and John and the Eastport Yacht Club. It's great to work with you.
And here's to a great session. Thank you all for your time, and thank you for joining us tonight.